In this session, we're going to have a few more talks. The next talk is called Single Cell Genomics Identifies Specific Cell Types and Pathways Affected in the Cortex of Autism Spectrum Disorder Patients. Our speaker is Dmitry Velmashev from UCSF. And he is going to... You're Dmitry, right? He's right here. He graduated with a bachelor's degree in biology in 2009. I'll give a brief overview here. From Moscow State University, started out in life studying epilepsy and Alzheimer's disease, and he's gone through quite a journey. Nowadays, he's at UCSF. He's currently a postdoctoral scholar. Focuses on applying single-cell genomics techniques, including single-nucleus RNA sequencing, to study cell type-specific mechanisms of psychiatric and neurological disorders, especially autism. Thank you, Dmitry.
Hello, everyone, and thank you for a great introduction. And I would like to thank the organizers for inviting me to give a talk today. So I'm a postdoc in the Kriegstein lab at UCSF, and what the lab really focuses on is on studying human brain development, especially cortical development. And lately, we've been using a lot of single-cell genomics techniques, try to profile different cell types during normal development and also in disease. So what I've been working on in the lab is trying to use single-cell nucleus sequencing to profile brain tissue of idiopathic autism patients. So I don't think I need to give too much introduction into autism. But just to say that it's a very prevalent disorder. It affects almost 2% of children in the United States alone. And what we know about the molecular mechanisms and cellular mechanisms is that there is a significant genetic component to the disease. But if we talk about idiopathic autism, it doesn't converge on just one or few genes. So there are hundreds to maybe thousands of genomic loci that have been associated with the disease. But what's interesting, despite this huge heterogeneity in terms of genetics and also in terms of behavioral manifestations of autism, previous studies identified shared and convergent transcriptional changes that take place in the brain of patients. But what hasn't been known before is hasn't been known too much before is the cell type specificity of these changes. So as I mentioned, there has been a lot of work done before using postmortem brain tissue samples and bulk transcriptomics, such as bulk RNA sequencing and bulk gene microarray, to profile the changes in gene expression that are associated with idiopathic autism. And overall, the main conclusion is that despite the heterogeneity of disease, there seems to be a convergence of changes on individual genes and also pathways. And some of these pathways include synaptic transmission and activation of microglia and astrocytes. But what we're really missing is a cell type resolution kind of analysis to identify specific cell types in the brain that are affected. So this is where the single-cell RNA sequencing comes in. Just the brief introduction to this technique is what it allows you to do is to analyze gene expression in individual cells isolated from a tissue. And what it can be used for is identifying cell types without the need of physically purifying them, for example, using FACS. And because of that, it can be used to identify cell types that have no reliable marker gene expression or rare cell types that represent a minor population of cells in the tissue. And it can also be used to analyze cell state transitions, such as during development or in disease. But one of the main caveats of single-cell sequencing is that it requires live tissue. So when it comes to neurological and neuropsychiatric diseases, in general, this tissue is not available. So what we have is a postmortem brain tissue. And I have a slide here just illustrating some advantages of using single-cell sequencing compared to bulk tissue RNA sequencing. So when you do bulk RNA sequencing, what you start with is a heterogeneous tissue which contains different cell types represented by different colors here. And you isolate total RNA, which is basically a mixture of RNA molecules from all these different cell types. And when you do sequencing, you can analyze the data in bulk. So if you have multiple samples, if you have samples from disease and controls, you can make some conclusions about the bulk total RNA quantities. And if you look at some hypothetical gene X, you might conclude that there is no change in expression of this gene. When you do single-cell sequencing, what you do is you basically barcode individual RNA molecules or populations of RNAs coming from each individual cell. So when you analyze the data, you can actually assign, you can identify how many original RNA molecules were in each specific cell or specific cell type. So one thing you can do is you can cluster these cells or cell types by how similar the gene expression profiles are. So it's an unbiased way to identify cell types without knowing the marker gene expression. And also you can identify a differential gene expression using this technique. And what you can actually conclude about the same gene X is that it might be expressed not in all the cell types you analyze, in specific cell types. And the differential expression associated with disease might be only restricted to one single cell type. So you get a kind of a better resolution of your analysis when you do this, when you use this kind of approach. So single nucleus RNA sequence is a modification of single cell RNA-seq, which allows you to profile single cell RNA profiles, not from whole cell, but from nucleus. And the reason why we want to do that is, for example, in our case, we obtained frozen brain samples from autism patients and controls, and there's no way to isolate whole cells because, first of all, it's postmortem brain tissue and it's frozen. But you can actually isolate intact nuclei from neuronal and glial cell types and use more or less the same techniques to profile their transcriptomes. So as I said, the advantage is that you can, using this technique, you can analyze transcriptional profiles of brain cells from disease samples on the single cell level. And what I'm going to talk about, and the protocol is mostly based on this paper from 2016. The main difference is that they use FACS to purify new and positive nuclei, so those coming from neurons and not glial cells. And they use this fluid MC1 platform, which basically captures nuclei or cells on a microfluidic chip. We use a commercially available version of this protocol called DropSeq, where instead of capturing cells and nuclei on the chip, you capture them in this microfluidic platform. So you can capture way more cells and nuclei using DropSeq-like methods. So the approach that we used, we looked at cortical samples from two different regions. And in total, we analyzed 41 samples from 16 controls and 15 ASD donors. So for some of the individuals, we analyzed both cortical regions. And in terms of the approach of collecting data, we have this small tissue samples from the cortex, and we first do serial sectioning of the samples on a cryostat to make sure that we collect tissue from all the cortical layers. Then we do nuclei isolation using ultracentrifugation, and then we use 10x genomics platform to capture single nuclei. And I should say that this has now been published, so if you're interested in more details about this study, this is the link to the paper. And as I said, using this technique, single-cell RNA sequencing in general, first thing you usually do is you cluster your data based on how similar the transcription profiles of individual cells or nuclei are. And then after that, you can annotate them based on marker genes that you know are expressed in specific cell types. So even though it's not driven by marker gene expression, you can use the marker genes to understand what these different classes represent in terms of cell types. So we were able to capture a number of subtypes of projection neurons from the cortex that are located in different cortical layers, as well as interneurons and glial cells such as astrocytes, oligodendrocyte precursor cells, mature oligodendrocytes, microglia, and also endothelial cells. And this is the marker gene expression that I mentioned that we use to classify these clusters. And just talking a little bit about the technical aspects of this data set. First of all, we know the identity of each nucleus, so we know if it came from a control individual or from an individual that was diagnosed with ASD. And also we just do this checks that we make sure that we run experiments in different batches. So, and this is known to affect gene expression. So we make sure that our batches are more or less represented in different clusters. So we don't have clusters only made of one batch or one individual. And also just looking at how many genes we can detect per cell or per nucleus, it's quite different depending on the cell type. So the highest numbers we can detect are in projection neurons and cortical excitatory neurons. And for glial cells, these numbers are lower. And I think it's mostly related to the size of nuclei that we purify. So probably the RNA content, for example, in microglial nuclei is much lower than upper layer projection neurons. And we also verified our clustering and cell type annotation using an alternative clustering approach. And just pretty much we can recover the same cell types and clusters, and they are pretty well correlated between these two clustering approaches. And before I move into the main part of the talk, which talks about cell type specific gene expression changes, I just want to mention that you can use this technique also to look at relative numbers of cells in different conditions. So you can normalize the number of nuclei per cluster or cell type, and then you can see if there's any changes in proportion of cells. So what we saw here is that there's an increase of protoplasmic astrocytes in the patients with ASD compared to controls, shown here. So this is just based on single nuclear RNA-seq data, and then we validated it using in situ hybridization. And then the main thing we wanted to do with this data is to look at gene expression profiles in the specific cell types. But before that, we wanted to do several more technical checks. So first thing we did bulk RNA sequencing of the same samples that we use for single nuclear RNA-seq. And what we wanted to do is to make sure that the nuclei profile, RNA profiles, are more or less correlated with the total RNA profiles from the tissue. And for most genes, it seems to be a positive correlation between nuclear RNA profile and total tissue, RNA tissue profile. And also we compared this bulk RNA-seq analysis from our patient cohort with previously published data sets using bulk RNA sequencing of ASD and control samples. And even though we have a completely different cohort of patients, I think there's only two that are common between ours and this published data set. We find a number of differentially expressed genes that are the same between these two studies. And if we look at whether these genes are up or down regulated, it seems to be a good correlation between them. So now moving to differential expression analysis in specific cell types. So once we have this cluster, so cell types, we also know which cell or nucleus came from either control ASD cohort, and we also know that there's other things rather than diagnosis that can affect gene expression, such as age of individuals, postmortem interval, RNA quality, and sex. So when we test for gene expression, we basically get cells from the specific cell type from each of these clusters, and we model gene expression as a function of these many different factors. But we're really only interested in the genes that change in response to this diagnosis factor, which is ASD versus control. But we control for all these other technical and biological covariates. So using this approach, we can identify genes that are differentially expressed in specific cell types in ASD. And this is just a volcano plot showing the differentially expressed genes either in neuronal cell types or glial cell types. And pretty much the take-home message here is that a lot of neuronal cell type-specific gene expression changes are coming from either upper layer projection neurons in the cortex or layer 4 projection neurons in the cortex. So upper layer projection neurons project to other cortical regions. So they're responsible for intercortical communication, and layer 4 receive afferents from thalamus. And looking at the glial cell type changes, it's mostly changes coming from either protoplasmic astrocytes or microglia. And a lot of genes here are actually upregulated. And looking at pathways, we pretty much find the classical autism-associated pathways, such as synaptic transmission and neuronal migration and neural adhesion. And one interesting analysis we were able to do here is to look at gene expression changes and how much they enriched in specific cell type. And technically, how we approach this, we have this clustering diagram for different cell types, and we can do differential expression analysis in each cell type. But the problem is that the number of cells is different between different cell types. So we have way more, for example, upper layer neurons captured than microglia. So in order to get rid of that effect, we downsampled each cell type to the same number of cells. So in this case, we would downsample it, I think, to the same number as the microglial cells. And then we repeated differential expression analysis to normalize for the cell number. And we repeated it 10 times. And then we were able to see which cell types are enriched for ASD-associated gene expression changes. And again, we see that this upper layer projection neurons come as the most enriched cell type. And it's really different compared to layer 5-6 projection neurons, which project to subcortical structures. And the other interesting observation is if you take all the differentially expressed genes that are differentially expressed in different cell types, and you cluster the cell types based on the fold change of gene expression, comparing the ASD and control, you can see the cell types cluster based on their developmental lineages. So you have excitatory neurons here, interneurons clustered together, and you have glial cells clustering together. So it kind of represents the way that it happens during brain development, even though we're analyzing postnatal adult tissue. Then we also see that we find a number of genes that are genetically associated with autism, so they have mutations found in patients. We can also do it by cell type, and it's mostly falls into the neuronal cell types. And I just give you several examples. It's a lot of genes, so I'm not going to go through all of them. But the main classes of genes that we see differentially expressed are synaptic genes and genes also coding for transcription factors that are important for brain development. And so these are for upper layer neurons and layer 4 neurons. And when we go into microglia cells, there's a lot of genes suggesting there is a change in the state of microglia, so perhaps increased motility as well as for astrocytes. And what's interesting, since we have two different cortical regions that we analyzed, we can actually look for region-specific differentially expressed genes. So there are certain genes that are only differentially expressed in the prefrontal cortex or the cingulate cortex in ASD. So the next analysis we wanted to do, we had some clinical information about the severity of behavioral symptoms of ASD based on this standardized test. So we wanted to see if the cell type-specific changes that we identified can be predictive or correlate with the clinical severity. So we performed this correlation analysis, and we just look for cell types, changes in which are most predictive of this clinical parameters. And again, we see that upper layer projection neurons correlate well with clinical severity, and we can actually even stratify the patients in different groups based on this is just based on gene expression changes, but it correlates with severity here. So those that have upregulation of certain genes or downregulation of certain genes in these upper layer cortical projection neurons tend to have more epilepsy, and many of them don't have speech development, proper speech development. And we can do the same analysis using the microglia genes as well. And the other thing we started to do, even though we don't have that many individuals here, we can actually, since we technically capture multiple replicates or biological replicates from the same individual, multiple single nuclei gene expression profiles, we can do analysis based on just individual nuclei just coming from that specific individual with ASD for each cell type. So what we did here, we compared the nuclei coming from each ASD individual to the average control profile in the same cell type, trying to look for differentially expressed genes that are specific to one individual with ASD. And they tend to converge on the same cell types when you do this individual level analysis, again, highlighting the role of these upper layer projection neurons, as well as these cortical projection neurons that are located in deep layers. So those are still projecting to other cortical regions, but they're located in layer 5, 6 of the cortex. And so there's a significant degree of comorbidity of ASD with epilepsy, and especially when you use in postmortem cohorts, this percentage is even higher. So what we wanted to make sure is that things that we're looking at are not just effects of either seizure or seizure medications. So we had a different data set generated from 8 epilepsy cases, 7 controls. So these epilepsy cases were not diagnosed with ASD or other neurodevelopmental disorders. So what we did, we performed the same analysis. We did single nucleus RNA sequencing, we did clustering, and then we identified gene expression changes that are specific to each of the cell types. And then we overlapped those cell type specific changes with the changes that we saw in the ASD. And overall, we saw a significant overlap, about 10% total differentially expressed genes are common between epilepsy and ASD data sets. And for upper layer neurons, it's about 20%. But overall, I think it's not dominating the things that we observe in relation to ASD. And if we take just the genes that are differentially expressed in ASD but not in epilepsy, we pretty much see very similar processes affected, such as synaptic transmission and neurodevelopment. So I'll just give you some conclusions. So first of all, single nucleus RNA sequencing can be used to profile cell type specific changes in ASD using postmortem SNP frozen brain tissue. And obviously it can be used for any disease where you have postmortem tissue from patients. And the ASD-associated cell type specific changes converge to specific cell types. And especially we saw an enrichment of these differentially expressed genes in upper layer projection cortical neurons, suggesting that perhaps these cortical circuits are specifically affected in patients. And as I said, they also converge, these genes also converge in specific pathways, such as synaptic transmission and neurodevelopment. We also saw that glial cell types also change their gene expression states, especially microglia and protoplasmic astrocytes. And what we started to do here is we tried to use this single cell genomics to analyze how specific cell types change in the brain of individual patients. And I mostly talk about idiopathic autism, but I want to say that we have a new project in the lab that we try to use similar techniques to analyze postmortem brain tissue from Dup15q syndrome patients. And we're combining this approach with 3D cerebral organoids that Stormy mentioned in her talk. And we're actually collaborating with her on this part of the proposal to use iPS cells derived from duplication 15q patients to model neurodevelopment in 3D. And we're still waiting to receive this brain tissue sample from Autism BrainNet repository. And the idea is to look at postmortem tissue to identify the effect of perhaps developmental changes resulting from this duplication, and also look at how brain development changes using this 3D cerebral organoid cultures. So I already told you about this approach. We have a number of people in the lab working the cerebral organoids. So they recapitulate certain aspects of cortical development, especially early stages. And we also use single-cell RNA-seq to profile different cell types in this organoid, since it's a heterogeneous cell culture system. And I would like to thank everyone in Kriegstein Lab, especially Arnold, and people that directly participate in this project, and collaborators, and the funding. This project was funded by Simons Foundation, as well as NIH. Yeah, so the first question is how we're gonna connect, I guess about the duplication 15q proposal, how we're gonna connect the development part of the proposal with the postmortem brain tissue. Yeah, I mean, there's a chance that there might be some genes that are common that are dysregulated in the postnatal tissue and during development. There are some cell types we capture in organoids, such as postnatal neurons. But it's more likely that the changes in organoids will maybe cause later changes that you see in the postnatal tissue. So we might need to use some kind of pathway analysis to see if there's changes that we see in organoids are upstream of what we see in the postmortem brain tissue. I don't know exactly how we're going to do that. We haven't started to work on this yet, but that would be like a general idea, I guess. And the second question is about 5 prime. Yeah. Yeah, the problem with we use 10x genomics, and it's you basically just have you get the reads from a short fragment of at the 3 prime of the transcript. Yeah, so you could do that. So in this data, we don't have that, but we could potentially run it in the same samples. I mean, we try to control for RNA degradation several ways. First of all, we cannot run this technique if the RNA is too degraded. So we had to design a certain cutoff for RNA integrity. So if it's below that cutoff, we don't even try to do that. And second, the RIN, which is RNA integrity number, is included in this model when we do differential expression analysis. So, yeah.
So can you do it on, I thought it was just on bulk RNA that you can get RNA integrity. Can you do it on nuclei?
Well, yeah, you can. But we basically what we do is we section the block of tissue that we get, and we use serial sections either for total RNA extraction or for nuclei extraction. And then from the total RNA, we get the RIN. Yeah, that's something we would like to do. We haven't done it yet. And actually, you can now do it on the single cell level to a certain degree. You can do attack-seq in single cells. So it would be really, I mean, it would be a logical thing to do next.
Maybe similar questions. So when you compare to the bulk RNA-seq, and the DEG genes is of less than 10%, so do you think that will be a significant number?
Can you repeat it?
Yeah, sorry. So that the compared to with the bulk RNA-seq,
yeah.
So the overlap is like less than 10%.
Yeah. So yeah, we did comparison. So yeah, we did comparison with bulk RNA-seq from the same samples, and the reason why we did it mostly is because we only get nuclear RNA profiles, so we don't know how well it represents the actual whole cell profile, and there's no way to get whole cells from this tissue. So what we did is we kind of made a bulk nuclear profile by just averaging all the single nuclear profiles for all the cells from the same sample, and then we did bulk RNA-seq from the same sample, and we just did simple correlation. So one thing we found is that if you just focus on genes that correlate very well between nuclear and total RNA, you kind of still see the same pathways like synaptic transmission. So if you only restrict to those differentially expressed genes that are well correlated, it's not a direct way, but that's the only way we can do it with this kind of tissue. I don't know if I answered your questions here.
So then the second one is, maybe I missed, but your samples 15, I don't remember the exact number. So what was the age of those patients?
The age was between 4 years and 20 years of age.
So the very different ages.
Yeah, different ages. We try to keep it like not older than 20. I mean, it's hard to get enough samples, but yeah, it was a range of ages.
And then all the mixed, basically.
Yeah, yeah. So the only way we did is to normalize for age is in our gene expression, different gene expression model, age is included as a confounding factor. So it's just like a mathematical way to model this effect. But yeah, we had to pull all the ages together.
So my question is partially related to previous one and your
Mhm.
answer. Related to, so doing the single nuclei RNA-seq, you obviously are missing what is happening in the cytoplasm.
Yeah.
So I understand that comparing the bulk and the nuclei, you can try to get an idea, but still it's difficult. Using different system like cells in culture, IPS, whatever,
Mm-hmm.
can you get a signature of what is happening just in the cytoplasm and not in the nuclei?
Mm-hmm.
To get an idea of what we're missing is like
Yeah.
RNAs that may be
I see.
being translated in the synapse.
Yeah.
Do you see those things being not, so do you miss that signature in the single nuclei?
Yeah, so we actually work with a lot with developing human tissue, so where we can actually get live cells. And we haven't done it yet, but what we're planning to do for another project is to compare just side by side, just get, use the same sample and do either single cell or single nuclei. And I guess maybe it's related to your question, there might be classes of genes that are more represented in nuclear, that correlate well between nuclear and whole cell profile. That would be good to focus on in this kind of studies.
And the other question is that, maybe I missed it, but I mean, obviously with 10x genomics, I mean, you have a very high threshold, so you're missing a bunch of
Yeah.
gene expressed at lower level.
Mm-hmm. Mm-hmm.
Do you have any way to follow up on those genes that you may be missing
Yeah.
in the 10x compared
So
to the
you mean
bulk?
like lowly expressed genes might not be represented there, yeah. I mean, one way is to use, we started doing it for another project where we did nuclear sequencing. We also did confirmation with, there was a study in multiple sclerosis. So we basically did the same, we used the same technique for NucSeq, but we also validated with a single molecule fish, like in situ hybridization. And it's better in picking up like more lowly expressed genes. And also 10x is coming, I think now there's a new version of the kit which is way more sensitive. We haven't used it for this project, but I heard that it's picking up more lowly expressed genes. So I think the technology is going to improve definitely. Yeah.
Cool, thanks.
Yeah, no problem. Thank you.
Thank you.
Thank you.